All right, let's go ahead and get started today. We are doing chapter 19. Don't forget, we do have an assignment that is due tonight on my Wartburg. If you forget how to find it, you go to coursework, and you will see chapter 22 is due tonight at 11:55 p.m. Your chapter 19 assignment, which is what we go over today, will be due Friday. All right, you will have an extra credit assignment. As you can see, it's due Friday, March 12th, so you have plenty of time to do that. It goes into your homework category, so it can get you a few of those points back that maybe you missed on some of those lecture assignments. On Wednesday, so a couple days from now, we'll be doing a lab, which we haven't had lab in a couple of weeks, but we're going to be doing DNA extraction. So if you go to the syllabus tab on our course, you scroll all the way to the bottom. All of our lab materials are here, so you're welcome to read ahead. We'll be doing DNA extraction on Wednesday, which you can see right here. And that one's pretty cool. You guys will get to extract and see your own DNA. That one's pretty fun. You'll remember that you'll be going in two groups. So remember the alphabetical groups that we have set up. The first half of the alphabet comes in right at 1:15. Second half of the alphabet comes in, we start around 2:10, but you guys are usually in there by 2 o'clock or 2:05. Okay. Any questions about that setup for the rest of the week? Cool. So chapter 19. All right, we're going to talk about chromosomes. Chromosomes, you may have heard of with different genetic diseases. Maybe one of the more common ones being Down syndrome. Common just because it's maybe talked about frequently. But what chromosomes are essentially our tightly wound DNA wrapped up on itself with some proteins to create essentially 23 pairs. And these 23 pairs are what makes us unique. Each location is a different trait. So what we're going to talk about today is that structure of chromosomes. What do they look like? How does their structure help with their function? And what is that function? We're going to talk about the cell cycle and what occurs at each of these stages. We know that most of our cells, but not all of them, kind of regenerate. Right? Obviously, you get hit in the head and you have a concussion, your brain cells don't regenerate, unfortunately. We have plenty of brain cells, but some things like your skin cells, your liver cells, your kidney cells, those are regenerating all the time. And the cells are at different stages of the cell cycle, and we'll talk about that here. We'll explain mitosis. We'll also go through meiosis. Those two I've written on the board a couple of weeks ago, maybe last week. Where we explain the difference between mitosis and meiosis, the difference in the spelling there, but also in their function and what they are for, okay? We're going to talk about some disorders of the chromosomes. We're also going to talk about some changes and what happens if there's some changes in the structure of the chromosome. So just a review, you may know that humans have 46 chromosomes, or essentially 23 pairs. And we're going to look at a picture here in just a second. And these pairs are called homologous chromosomes. Okay, just to give you a brief picture of what that means, I'm just going to skip ahead to a couple of slides. These are the pairs of chromosomes. So as you can see, there's a number 1. Technically, these are 2 chromosomes, but this is their matching pairs, okay? As you can see, they're pairs because of their size. So you can tell that these are the same, these are the same. In a class that I've taught at UNI, they would throw a bunch of these chromosomes onto a sheet of paper and they'd have to match them. And you can match them because each of them has a pair, okay? That's called a homologous chromosome. It's made of a matching pair. All right. The autosomes are the 22 pairs of chromosomes that control traits that are unrelated to gender. Okay. and by gender meaning just the X and Y chromosomes. So when we talked last week about autosomal dominant and autosomal recessive, this word autosomal or autosome, we're talking about the same thing here, okay? This word right here in red, autosomes. We talk about autosomal inheritance. Autosomes just means all the regular chromosomes besides the sex chromosomes. And then you have the sex chromosomes, which are the pair that contains the genes that do control gender. So in that location, on the 23rd pair, you'll either see an X, a Y, an XX, or we're going to talk about some disorders or some things that are a little bit more rare, where maybe there'll be 3 X's, or maybe there'll just be 1 X, or maybe there'll just be a Y. But there's other disorders in that location that may lead to a genetic disease or disorder. But most of the time in a normal individual, you'll see an XX or an XY. The words diploid and haploid are ways that scientists describe the types of chromosomes that this individual has. So while it's easy to think that humans are the only things with chromosomes, that's not necessarily true, okay? Other animals, other organisms, even some microorganisms that have DNA have chromosomes, and not all of them have pairs like we do. So it's easy to get into thinking that all chromosomes look like human chromosomes, but that's not the case, okay? A haploid and a diploid are how many pairs that the chromosome has. So cells that have 46 paired chromosomes are called diploid. So that's what a human is. A human is a diploid because we have 46 pairs, or 2N is another way to say that. The N being the number of pairs that we have. You can also have an organism that is haploid. A haploid organism is one that is unpaired, that would just have 23 single chromosomes. Not like that picture that I'm going to show here on the next slide. A karyotype is a word that describes a picture of chromosomes. So this is a karyotype. Okay, it's a picture that was taken of some chromosomes so that we can look at them. A karyotype, okay? I like the little description here on the right. So each chromosome has a pair, in this example, called a homologous chromosome, or a homologous autosomal pair, okay? This is a homologous autosomal pair, right here, right here. All of these are homologous chromosomes. And then within this one strand here, we have sister chromatids. So we have sister chromatids, homologous pair, and then in the middle there's this thing called a centromere. A centromere kind of to me just looks like a little knot. Like if this was made out of string, it looks like a little knot that holds the chromosomes together. It holds the sister chromatids together. The other thing we can notice from this picture is although all the chromosomes are numbered, we could tell whether or not this individual had a genetic disease and whether or not this individual was male or female just by looking at the picture, okay? But in this case, the picture is numbered, which helps us. But if I were to kind of cover up this X and Y, you could easily tell that this individual is male, because if it was an X, these two in the 23rd location would be the same. So this is an X and a Y because look, the Y is much smaller, okay? But if it was XX, it'd look like this and this. It'd be the same. So that's a way you can look at this picture and figure out the sex of the individual. You could also look at each of these locations and see if there's any extra pairs. Maybe this one would have an extra pair. And then in that case, we would identify what that disease means. But just because it's numbered and labeled doesn't mean that we can also see things when it's not numbered and labeled. Okay, so that's a little bit about chromosomes. Okay, and to give you an idea of where our chromosomes are located, I like to draw this very nice drawing. Okay, so we know that our DNA is double helix, right? This is DNA. We worked with that last week, right? DNA. Good. Now, your DNA then is tightly wound together with chromatin. Chromatin is a mixture of DNA and protein. So I'll say chromatin equals DNA plus protein. Essentially, think if I just tightly wound a couple of pieces of string together, really twisted them together, and then what that ends up looking like are your chromosomes. Okay, so this is kind of like a blown up version of what your chromosome is. So it's a tightly wound DNA. The reason why chromosomes are just tightly wound DNA is I like to give this example, and I always hope that the classroom I'm using has a piece of paper. Let's see, does that have a piece of paper? I'll use a piece of notebook paper for my example here. Okay. So I've got this piece of paper. Let's say that this is the DNA, right? And I wrote some code on here. All right, maybe I wrote A, G, C, T, G, 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 C, C, C. All of this is our DNA, okay? And I wanted to be replicated and divide so that I can pass this on to my next cell or my offspring, right? This is really important stuff because I'm, the best organism out there, right? So I've got to pass on this very nice DNA, okay? And I just go to get replicated. I probably damaged it, right? Because my paper was wide open. All right, another way to look at this is if I tightly wound this DNA, I'm protecting the message on the inside, right? You guys probably, I didn't write anything on this, but now you can't read the message on the inside, not unless you had some enzymes or something. And it's going to be a little bit more protected, and I can throw it a little bit further. Okay, so not necessarily that we're throwing our DNA in our cells, but what I want you to know is it's tightly wound into these things called chromosomes. I didn't even, sorry if you, that was a little aggressive. This DNA is tightly wound with DNA and protein into our chromosomes to protect it for when we are dividing and replicating, when we want to make a new cell. Okay, so it protects it, and it does allow it to move a little bit more efficiently. Remember I told you that if we were to unwound our DNA, the small little string would be about 6 feet tall. Okay, that would be really hard if that was just our DNA, and every time we needed to make a new cell, that thing would have to replicate itself. Okay, so that's why we have these chromosomes that are tightly wound DNA and chromatin. Okay, and it's to protect it. That's why it's really kind of wound up like that. And also for when it divides and replicates, it can essentially move a little bit better, but we're not throwing it like a piece of paper. So now our goal is to make a new cell. Okay, all of our cells, besides like our brain cells and some nerve cells, are regenerating constantly. They're also at different paces. Okay, remember when I say that we have, we get new cells and our skin cells regenerate every 2 weeks. We're not a snake, right? We don't just shed it all off at one time. Every cell is at its own stage. Okay, and so our goal here is to determine what are those cell cycle stages, or when we're going through mitosis, where are we at in a cell cycle stage? Okay, our goal when making a new cell is to replicate our DNA that's on the inside so that we have another copy of it, and then split in half. And then from those cells, we replicate the DNA on the inside and then split in half. Okay, we make a copy and we share. We make a copy and we share. All right, so the first step here is called interphase in the cell cycle. And there's interphase, and then there's these things called G1 stage, S stage, and G2 stage. I picture those as like a little kind of checkpoint along the way. So when you're going through interphase, the cell doubles its organelles and the cell grows in size, which makes sense, right? I have an animal cell, pretend I'm holding an animal cell, and I want to make another animal cell. So if I need to make another one, all those organelles that are in the first one need to come over to the second one. In the S stage, our DNA is replicated, all right? DNA is kind of in charge, right? They're like the CEO of your cell, okay? And the DNA replicates it so that there can be a DNA over there. You got to have a new boss in the new cell, all right? And then we have the G2 stage, which are proteins that are needed for division are synthesized. So we're going to have the function of breaking these two cells into two pieces. And so proteins are going to kind of be in charge of that function, okay? Most cells are in interphase. If we were to look at a slide under a microscope of a bunch of your cells, let's say it's a bunch of skin cells, and I said, determine where they're at in the cell cycle based on, what their chromosomes are doing, about 70% of your cells are in interphase most of the time. And we'll see that, I think there's a picture. Yeah, on the next slide. You see that big purple arrow? That's interphase. That's what essentially your cells are in most of the time. Like I said, it's about 70 to 75% of the time. Okay? Cell division is then what happens next. So in interphase, what we did is we created some more organelles, we created some more DNA, we got some proteins that are going to help us pull them apart and be super strong, and now we're going to divide. And we go through a division called mitosis. This is a nuclear division, which means that we're dividing the nucleus. And then there's also a division called cytokinesis, which is where we divide the cytoplasm. The cytoplasm is that space where all the organelles are kind of floating around. It's kind of like the pool where all you hang out and then all the people are the organelles. Okay? So when you look at this next slide, I like this picture a lot. Okay? So we have interphase, which was that first step. I told you this is where your cells are most likely in right now because there's a lot that has to happen. It's not that the growing and dividing happens quickly, but it also doesn't happen super slow. It's kind of hard to explain. So there's this growth stage, and as you can see, there's kind of a little bit of a feedback loop here where your cells may stay here for a while. An instance like this would be a fetus growing and developing body cells and things like that. We're going to have to stay in this growth phase a lot. We're going to have to grow, and then it goes into the S phase, where we grow the DNA and replicate it. And then after that, we make some proteins and prepare for the division. Okay, once we get to this checkpoint here, we go through mitosis. Mitosis, let's write it on the whiteboard. I like whiteboard better than chalk. Anybody else? Oh no. Whiteboard over chalkboard. Yeah, that doesn't make that screechy sound. Ooh, don't like that. Okay. We'll use chalkboard. Okay. Okay. So mitosis. The other one we're going to talk about here in a little bit is called meiosis. Okay, the way that I want us to remember mitosis is like my toe, which is silly, right? But all of our cells that are not our sex cells go through mitosis. So this is your skin cells, your hair cells, your liver cells, your kidney cells. All of those things are going through mitosis. Okay, it has replication because we want to make sure that we have enough DNA, and it has one division. And what we end up with is two identical daughter cells. Okay? So this phase right here is called mitosis. Now in our class, I don't have you memorize all of these steps, but you maybe had to in high school. It was like prophase, interphase, telophase, metaphase. All of those, we don't memorize them. Just kind of understand, and we'll see here in a couple of pictures that the chromosomes kind of come towards the middle and they divide after replicating and things like that. After this, The cell goes through cytokinesis. This mitosis phase is called nuclear division, or dividing the nucleus so that the new cell can also have a nucleus. Cytokinesis comes next, and that's where it's dividing of the cytoplasm. Okay? There are checkpoints that can delay the cell cycle until certain conditions are met. So these checkpoints happen kind of in interphase. G1, if it's passed, the cell is committed to divide. At the checkpoint G2, the cycle pauses to verify that the DNA is replicated, which makes sense. We don't want to just keep moving uncontrollably and then make a mistake. We got some precious cargo there, right? This is the most important DNA ever. So M cycle pauses to verify that the spindle assembly and the chromosomes attachment. So G1 happens in the first G phase. There's a little stop sign. You guys see that stop sign? Those are the different checkpoints. G2 down here is not just Gatorade. It also is a checkpoint where we're making sure that those proteins were created so that we can divide appropriately. And then that M checkpoint is when mitosis will not continue unless the chromosomes are not properly aligned. Okay, so I think of these also just kind of like we had that airport example last week. This is kind of the same thing. It's like the different security checkpoints that you go through in the airport before you can board the plane. Okay, first, you got to stop and show your ID and show them your boarding pass on your phone or check your bag. That's probably like your first stop, right? They're going to stop you a couple of times before you get onto the airplane to make sure you are who you are, right? And that you don't bring anything suspicious on through your bags. That's essentially what's going on right here too. We've got these different checkpoints. They're kind of like security points in in the airport, or maybe before you go into a large concert, because we do that all the time nowadays. And you get all of these different checkpoints, and once you've kind of reached those criteria, you can keep moving forward. You could also think about it like in school, right? You had to have certain graduation requirements in high school, and once those requirements were met, you could move on to the next step, right? Now you're in college, you probably got some certain graduation requirements before you can graduate college, and once you hit those steps, then you can move on to the next part. And then maybe you're applying for a job, you have to submit an application, and on the application you have to have a resume and a cover letter and whatever, all of these things, references. And then once you get all of those things done, you can turn in your application. Those are like what those checkpoints are for the cell cycle. It's important because it helps to make sure that we are limiting mistakes, okay? Because mistakes happen, mutations happen in cells. They're rare, but we want it to be good a majority of the time so that your skin cells look like your skin cells all the time. An example of when something would kind of go wrong there is like in skin cancer, right? You're growing and dividing of a cell that maybe has a mistake in it, or it's uncontrollably dividing and you can't stop it or control it. That's kind of where we get into play with like skin cancer or tumors and things like that, is when we're kind of having this uncontrollable growth of cells. Okay, so chromosomes contain both DNA and proteins called chromatin, which we already went over on the board. These chromosomes that are dividing are made up of two identical parts called sister chromatids. We talked about that a couple of slides ago on that picture. And these sister chromatids are held together at a region called a centromere. I kind of think of the centromere like if you tied a knot in two pieces of rope. It's kind of what's holding the two pieces of rope together. Here's another example of picture of that, where you can see the chromosome, and through replication, we get sister chromatids, and through division, we just get a single chromosome. So that's how you kind of work back and forth in this picture is, here's a chromosome, and when we replicate, it looks like this, and it has a centromere, and it has a sister chromatid, and it's a duplicated chromosome, but when I want to divide that, we go back to a chromosome. There's this word that was thrown around a couple of slides ago called the spindle. I think of a spindle like in Sleeping Beauty. I think that's what a spindle is, when you can kind of push your thing on the floor and then the wheel turns and then she pricks her finger. Okay, so a spindle fiber, okay, we also call these in a cell, a spindle fiber is what's going to pull those chromosomes apart in division. They kind of look like these little strings, and they kind of are because they're fibers. So these spindles in mitosis, they use the centromere, which is that knot in the middle of the sister chromatid, the microtubule organizing the center of the cell so it can, kind of organize all the centromeres together. The aster is an array of microtubules at the poles, and we're going to show a picture here in the next slide, so don't get too excited here. And then the centrioles are the short cylinders of microtubules that assist in the formation of these spindle fibers. So what we're trying to do is produce these little string-like fibers that are going to pull the chromosomes apart after we duplicated them to make two identical daughter cells. Our goal here is to replicate and divide and make new cells. Okay, so here's an example of those fibers. Right, my fingers were doing a really good job of demonstrating that. So we have the chromosomes in the middle. Those are those blue and red kind of X looking like things. The centromeres are the little dots in the middle that are essentially the knots that hold the sister chromatids together. You can see that the spindle fibers are attaching to the centromeres, or the centrosomes, and that the aster are these other kind of fibers that are attached to the outside of the cell, and they're going to just kind of pull those chromosomes apart. They're going to pull those sister chromatids apart. What an overview of mitosis can tell us is that in a diploid cell, so in the cells that are like humans where we have 46 chromosomes or 23 pairs, it divides an exact copy of its nucleus. Like it says, we end up with two identical daughter cells, okay? This is used for cell growth and repair, okay? You get a little cut on your finger, your skin cells are going to figure it out, mend the finger. Yes, you may have some scar tissue, but there are cells being created there. There are 4 phases: prophase, metaphase, anaphase, and telophase. And this occurs in all body cells except for your sex cells. Here's another overview of mitosis. Remember, it's a nuclear division. We're dividing the nucleus, okay? Here we have at the top, you can see a centrosome, a centriole, and the chromosomes up here at the top. We go through DNA replication. So as you can see, the cell on the other side of that arrow now has sister chromatids. They are duplicated chromosomes. We replicated or made a copy of chromosomes. And then we use those spindle fibers to pull at the centromeres. And now down here, we created 2 identical daughter cells. What that means is that if you look at this cell right here and look at the original cell, they should be identical. This cell right here and this one down here are identical. But we started with 1 and we created 2. It's not really like when you have a mom and the mom has 2 kids. That's not necessarily what's going on here, because the original cell is still a part of both of these cells. It's not like it still exists, if that makes sense. That's kind of hard to think of. But when somebody says 2 identical daughter cells, it's not like the original cell had babies. If that makes sense. That first phase of prophase, we're going to break it down here a little bit. Remember, you don't have to memorize the order necessarily of like the names, but you'll have to understand the gist of what's going on, okay? Chromosomes condense and become visible, okay? So I talk about how they're super tightly wound and condensed, okay? When they're normally kind of in an interphase and they're growing and they're replicating and things like that, they aren't as condensed as they are when they're going through prophase, okay? The nuclear envelope fragments or becomes open, essentially. So that nuclear envelope is what encloses the nucleus, and the nucleolus disappears. The centrosomes move to opposite poles. So these are the things that are going to pull those chromosomes apart. And those spindle fibers start to appear and attach to those centromeres. Okay, so those words that maybe kind of got a little confusing there were centrosome. I don't know, those kind of look like little churros, don't you think? Kind of look like a little churro. So those little churros, or the centrosomes, have these little fibers on them. That's what starts to be visible in prophase. Here's another picture of that, okay? This is a cartoon picture, and then this is like a real life picture of a microscope that's showing it, okay? You can see the chromosomes in the nucleus here at the top. You can see the aster and start to see the centrosome. Those are those fibers right here. You may want to look at this one in your book when we're done with the PowerPoint because projecting it is not as great, okay? You can now see that there's duplicated chromosomes. You know they're duplicated because that's the normal amount, and now they're super condensed into them dark, okay? The duplicated chromosomes, you can see the centrosomes, you can see these fibers that are kind of attaching there that are going to pull that apart. Remember in prophase, we can see the condensed chromosomes in the nucleus, and we can start to see the fibers, the spindle fibers. So you have that early prophase, and then kind of like the main show of prophase. The next one is called mitosis. Does anybody know who Meta World Peace is? Ever heard that name, Meta World Peace? Okay, I don't really know a lot about Meta World Peace, but when I say that out loud, I just think of people like coming together, like world peace, right? Meta World Peace is another way to remember metaphase because all the chromosomes are going to meet in the middle, which I think is kind of a nice way to remember that. So as you can see here, the metaphase, all these chromosomes kind of line up in the middle, kind of like the start of a sporting event when all the starting players and the bench or whatever gets to stand up and gets their name announced. This is that time, right? The chromosomes are coming up, they're lining up in the middle, or the equator, I guess, of the cell is what they say there, and the spindle fibers become fully formed. We're getting ready for the main event here, all right? So essentially when I think of like world peace, they like meet together in the middle. Then we've got anaphase, okay? The sister chromatids are pulled apart to start to become daughter cells, okay? So now you can see the chromosomes are kind of like this because they're getting pulled apart. You can see that they're like this. And they're being pulled apart at their centromeres because we're going to create two cells out of this, okay? Starting to look a little bit more like a butternut squash. And the centrosomes move towards the poles, or those centrosomes. The way I remember anaphase, which is kind of weird, my sister's name is Annie. This is really far-fetched. My sister's name is Annie, and the sister chromatids are pulled apart. So sister, Annie, I don't know. You can think of your own way to remember this one. All right. Then we have telophase and cytokinesis. All right, so we're getting here towards the end, right? We just separated our chromosomes, but what we need is to make this a little bit more official, right? We're about to separate these two into two individual daughter cells. This thing is called a cleavage furrow. You can understand how we can remember that one really well. The chromosomes become indistinct chromatin again. The nucleoli appear. Spindle fibers are going away. Nuclear envelope reassembles. We're kind of doing like a cleanup crew here, right? We've got everything kind of lined up, ready to go. And then these two daughter cells are formed by a ring of actin filaments and that cleavage furrow down the middle. So we're about to split these into two different cells. We can see that now those chromosomes have a nuclear envelope and a nucleus that they're housed in. So a little overview of what we just went through. There are two nuclear divisions that occur to, oh, whoa, I thought that was a review of mitosis. Never mind, that's meiosis. Does anybody have questions about mitosis before I move on? Mitosis. I guess the other thing that we can add here is that it's your body cells. You can read my handwriting from far away. These are your body cells. Okay, so now we're going to go on to meiosis, which is another kind of great exam or homework question. It's like, describe the differences and similarities to meiosis and mitosis. Okay, it's the same one of like, describe the similarities and differences to DNA and RNA. This is just one of those that you can't help but have on the test, right? So we've got in meiosis, these are what makes our sex cells, okay? Which is kind of hard to think about sometimes, that sex cells are created. And the way that I remember this one is me. Like, how was I created? I was created from two sex cells that came together and that were fertilized, and then they're growing, dividing through mitosis. But at one point, these sex cells had to be created. And we know that through the cell theory, because cells are not created or just created from thin air. They're created from other cells, okay? And it's different in males and females, where females, while the eggs mature over time, females are born with all of the eggs that they will have in their life. Males that produce sperm, that is produced kind of after puberty. They say throughout life, but it decreases into later age, okay? So meiosis is, how do we create a sex cell? How do we create an egg? How do we create a sperm cell, okay? And our goal here is to make that sperm or egg cell have half the number of chromosomes that we have. We want it to have half so that when the sperm meets the egg, each of them have half and it makes a whole, okay? You can get all sentimental about that if you want, but you don't have to. It is used to make gametes. Another word for gametes are the egg and sperm. Meiosis occurs in sex cells, like I said, and it has two nuclear divisions. So what's really cool about mitosis and meiosis is the first few steps are exactly the same. They're pretty much the same. We want to duplicate our DNA or replicate it or make a copy of it and then divide it and make two cells. That's the first steps of mitosis. Like if you remember from before, this looks very similar until about here, right? We have two divisions because from the two divisions, we can create now half the number than what we started with. And now these are individual, say, egg cells. They could be a sperm cell, I don't know. Okay? It has 8 phases. So the first thing that this has is also replication. Just like mitosis. But what's unique is that it has 2, I'm going to write that really big, divisions. Right? We want to end up with half the number of chromosomes, because if we had eggs and sperm cells that had 46 chromosomes in them, you'd combine them together and then it'd be what, like 95? I can't do that math right now. What's that? 90? What's 46 plus 46? 98? No, 94. 92. There we go. See, I'm a biology major, not a math major. All right, that's a lot of chromosomes. We don't really want that necessarily. We want to have offspring that look like us. That's kind of the goal. Okay, so meiosis, here's some steps. If you remember from the mitosis, we got the same names. This is super helpful. We got prophase, metaphase, anaphase, and telophase. Okay, the homologous chromosomes. Then also there's this thing called crossing over. Do you see that up there? Crossing over occurs. What is crossing over? Do we have a slide about crossing over? Yes, we do, in a couple of slides. So hang on to your seats, and we'll talk about what crossing over is. It's where the exchange of genetic information happens. It's also where we get some variety in our DNA, which is how you look different than a sibling, or how you honestly have a little bit DNA different than a twin sometimes. The metaphase 1 is where they line up at the equator. Remember, meta world peace. Anaphase 1 is where they separate and move towards opposite poles. Telophase is 2 daughter cells that result, and each have 23 duplicated chromosomes. So this is exactly the 2 daughter cells. This is where we ended at mitosis. We're doing the exact same thing. Is that a train or what is that? Mm-hmm. It's a train? Nice and loud. Okay. Then we can go through a couple of the pictures here. So this is for a diploid cell. Prophase 1, chromosomes duplicated, homologous chromosomes pair during synapsis, and crossing over occurs. As you can see, the crossing over right here kind of looks like a braided Twizzler. This is essentially where the chromosomes kind of exchange DNA within themselves. They kind of cross over, and it's what makes the genetic variation for offspring. And it's our benefit because it's also the question, why don't we all just look exactly the same? You know, there's some benefit to that, right? So over time, right, there's this thing called fitness, which you think about it with animals, right? If I'm a tree frog and I'm bright red and I'm redder than all my other tree frogs, all right, I maybe could be seen as more poisonous and I could survive longer, okay? Or maybe there's a benefit to being the brownish colored tree frog so I can blend into my environment, okay? So the reason we all don't look the same is because there's fitness and variation through crossing over in our genetics, and that there are some traits that have carried over time, such as that like humans are getting bigger, we're taller more now so than we were 100 years ago. I say we, because we're humans. I've been the same height since 8th grade. And then there's other things like different diseases that maybe we're more resistant to now because of either vaccinations or they're just not as prevalent, things like that. So anyways, crossing over is important for genetic variation, and genetic variation is important so we don't all look the same or have the same traits. And because we don't all look the same and have the same traits, there's benefits to that, okay? It's not just saying that if you look a certain way, you're gonna get, you know, more McDonald's than the person next to you. It's just a little bit more on the genetic level. All right, now we have, let's see here. I clicked ahead on this one, but not this one. Then we go into metaphase, which is the same as the other metaphase where they meet in the middle and we're gonna get ready to split apart, right? They're gonna kind of line up in the middle. As you can see, on the right-hand side, the chromosomes just look a little different. Like the blue one has a little red kind of part on it, like a little tail, and the red one has a little blue tail. And these two kind of look a little bit different. That's just showing, in a sense, that crossing over occurred and that now there's some genetic variation between the cell that we started with. Then we have anaphase, where as you can see, the chromosomes look like this, and they're getting pulled across, and they're going to be ready to be separated. Then we have telophase. We're literally so close to splitting in half. We've got that cleavage furrow right in the middle. We're going to split in half here. This is meiosis 1. That was one division, right? Because we just divided into 2 cells. This is exactly the same steps that happen in mitosis, minus the crossing over, the genetic variation. All right. Let's skip ahead to what is crossing over. Like I said, here's that slide describing it a little bit more in words so you can see it. It's the exchange of genetic information between non-homologous sister chromatids. So like it's where the blue one exchanged with the red one. Those are homologous pairs each, right? And this occurs during prophase 1 of meiosis, and it increases genetic variation. Genetic variation is good. If we all looked the same and we all did the exact same thing, there'd be some sort of competition for like our food. Like think of it more like we're animals, right? Because newsflash, we're all animals. If we all were birds and we all were black birds with the same beak and we all could only eat a certain type of food, it's just not good for the environment. We're going to lower what that food is because we're going to be eating it a lot, right? We're going to have some competition between us for that food because we all look the same. So there's just some benefits to being genetically varied, at least from a molecular level. Here's a picture of crossing over. Like I said, you can kind of picture it as like a braided Twizzler. It kind of looks like a nerd rope, kind of. I'm getting a little hungry for some candy. The sister chromatids are right here, And as you can see, it also helps that this picture has some letters on it. So you can see like the big A and the little a and how they are up here, and big B's, little b's, big C's, little c's. It just increases genetic variation. There's this other thing that increases our genetic variation. It's called independent alignment. So if the question asks, what are some ways that genetic variation can occur in sex cells? One of them would be crossing over. The other one would be independent alignment. Independent alignment is essentially how are we dividing those chromosomes. In a lot of these examples on the pictures, they divide them, that's visually pleasing, where they like put the reds on one side and the blues on the other. And the type A person in me is just like, thank you. I like that they're color coordinated. The size is perfect, but that's not necessarily the case. This is how those homologous chromosomes are going to line up in the middle, because they could line up in different ways, and how the maternal or the paternal member may be oriented towards either pole, and it gives rise to various combinations of maternal and paternal chromosomes and gametes, and it descends from the cell. So essentially, how are we dividing and lining up and being pulled apart to divide? Because that can kind of create some genetic variation too. So here's a picture of that, which really helps to show you some different variations of how they lined up in the middle. So you can see, like maybe all the blues line up on this side and all the reds line up on this side, and we divide. Okay, your chromosomes aren't really blue and red, but just to show you that there's different types and sizes of chromosomes. Maybe this one was 2 blue and a big red, and 2 little red and a big blue, and they divide. Okay, here's another example and another example. All of these are different combinations in which all they did was line up in the middle and divide, and because of the way they lined up is what changed. You know, if we think back to like a sporting event or maybe a music concert when somebody's going to stand up and be announced before what they're doing, okay, it's essentially like changing the starting lineup or putting somebody in there that's different and then seeing how it goes. That's a way to think about that. So that's how another way that genetic variation can be found in meiosis. So we got crossing over, where they kind of twisted together and created a different pair. Then you have independent alignment. How did they line up and divide? That gave us some genetic variation. Okay. Now we're on to the second stage of meiosis. This is different than mitosis, because mitosis we only had essentially one process and then we ended up with 2 identical daughter cells. But we know in meiosis that we want to end up with 4 genetically varied half chromosome or haploid individuals. And I say individuals, but individual cells. Okay, so we're going to go through it again. So from those 2 identical daughter cells, we're then going to condense the chromosomes again. We're going to align them up at the equator, separate them to opposite poles, and then we result in 4 daughter cells that have 23 single, ready to mingle, unduplicated chromosomes that are half the number that we started with, and they're genetically variant. Like they are different than one another. With mitosis, they were identical. They were just like a split of the exact same cell. So here's mitosis 2. We've got a picture of prophase 2, which is essentially the same as prophase 1, but it's happening in 2 cells at the same time. Okay, where you condense the chromosomes, you replicate them, getting ready to divide. As you can see, we're still dealing with 2 cells here. At that stage of metaphase, we now line up in the middle, getting ready to separate. As you can see, we have some genetically variant chromosomes. Like I said, we have like a red one with a little blue part right here, blue one with a little red part. But then we also have these ones over here that are also genetically variant that are happening at the same time. We then get ready to kind of pull those sister chromatids apart, as you can see, and now they're genetically different than what we started with. And in telophase, we're getting ready to divide them. We have that cleavage furrow, we have a nucleus, the spindle fibers disappear, the nuclei forms, cytokinesis takes place, so we're dividing the cytoplasm, and we end up with 4 haploid daughter cells. and they are genetically, I'll just say different. You could say unique, genetically varied. I'm just going to write genetically different. They're different from one another. Okay. The word spermatogenesis is just the process of making sperm in males. So the purpose of this next slide is just to show how do we make sperm and egg cells from meiosis. Okay. This is a continuation after puberty, which we talked about in males. Okay, so meiosis is occurring in males continually after puberty, and about 400 million sperm are produced per day. Now remember, the sperm don't have personality. Their only job is to grow, reproduce, essentially make copies of the genetics and go fertilize an egg, right? It's not a conscious behavior of anything except for that they think that their genetics is the coolest and they want to pass it on, okay? That's why 400 million sperm are produced per day, is because it increases the odds. That's why so much is made. But then we've got, in females, oogenesis, which is the process of making eggs in females. This is during meiosis, one egg and 3 polar bodies are formed. You don't really have to remember that as much, but remember how we end up with 4 haploid daughter cells? Essentially only one of them is really good, okay? The polar bodies act to hold discarded chromosomes and thus disintegrate, okay? Normally 1 egg per month is produced, and about 500 are produced during the entire reproductive cycle, okay? So this is like post-puberty and before menopause. It's different in every individual female. It could last longer or shorter than other individuals. Eggs can be different in individuals and things like that. So that's a little bit about oogenesis. So we have sperm and egg cells reproduce. So the difference between sperm cell production and eggs production is the time in which it happens. So the egg cells are already in the female body when the female is essentially born, but the egg cells mature after puberty. And then for sperm, the sperm are not necessarily within the body of the human male when it's born, but after puberty, sperm are produced every single day, about 400 million per day. Here's another little example of spermogenesis and oogenesis, just to compare, okay? You can see the comparison, how this looks a little bit like meiosis, right? We have the 2 divisions, 1, 2, in the divisions, okay? And then you see down here, meiosis 2 is completely after entry of the sperm, which is kind of interesting. Yeah, there's a lot of trains today. Okay, this is another kind of what we wrote on the board. Okay, we've been doing this as we go along. So as you can see, this is a good, maybe a Venn diagram studying tool, a good exam or homework question. What's the difference between mitosis and meiosis, and what are similarities, and why is it important? That's a big one, right? Why is it important? So mitosis, the growth and repair of cells, is going on as we speak. Okay, this is all of your body's cells. There's only 1 division because we want to create 2 identical daughter cells, and it results in 2 diploid genetically identical cells, which is what we want. Meiosis is the formation of gametes. Another word for gametes is sex cells, or sperm and egg cells. Like it says, it occurs in sex cells. It has 2 divisions because we want to create half the number of chromosomes. We want to result in 4 haploid genetically different cells. Here's a picture comparing mitosis and meiosis. Man, you must really think that this is going to be on the exam if there's been like 10 questions about it, right? Or 10 slides, all right? You can see that the first few steps of meiosis 1 and mitosis are the same, but where they differ is that mitosis is just 1 division. You end up with 2 identical daughter cells. Meiosis is different because it has an extra division, and you end up with 4 genetically different haploid daughter cells. All right. So now we can talk about when there's changes in chromosome number. Okay, so sometimes the growing and reproducing of these sex cells produces a sperm and egg cell that maybe has something a little bit off. Okay, when I see the word non-disjunction, it literally says non and dis. To me are both like negative connotation words, so we know something's a little off here, right? So non-disjunction occurs when both members of a homologous pair go into the same daughter cell during meiosis 1, or when meiosis 2 happens and the sister chromatids fail to separate. So essentially, you're thinking of like when we divide during meiosis 1 and they all went to the same chromosome now, or the same cell, now we've got some extra chromosomes in one and not enough in the other. Or in meiosis 2, when the sister chromatids fail to separate at all. What can happen here is something called monosomy and trisomy, okay? The word mono means one. Like when somebody says, let's take it outside, mono, right? That just means one-on-one, right? So when you see the word mono, it means one, and this is when the cell has one copy of the chromosome. An example of this is Turner syndrome, when there's only one X chromosome. Remember at the beginning of class, I said we can look at a karyotype and tell if it's a male or a female, because if it's a male, the chromosomes in the sex chromosome area are going to be like a big one and a short one. If it's a female, they're going to be the same. Well, if you have Turner syndrome, you're only going to have one X chromosome there. Trisomy is an example where there's 3 copies of a chromosome in one location. Tris, or TRI, like tri, like a triathlon, has 3 events. So this one has 3 copies of a chromosome. An example would be Down syndrome, when there's 3 copies of the chromosome on location number 21. And honestly, Down syndrome is fairly common. Common in a sense that we talk about it, and you maybe know somebody who has Down syndrome or a friend who has Down syndrome. It's definitely, I would say, a little bit more common than Turner syndrome. So here's an example of non-disjunction, so you can see what I mean when they're kind of just growing and dividing in different ways and it's just not working out, okay? So what happens here is that they all went during meiosis 1 and they split all into one side, okay? What that occurred with is then we have extra chromosomes here, which would be trisomy, 1, 2, 3, and then also end up in monosomy, or mono, like the MONO, where there's only one chromosome in that location. Non-disjunction. We split in a way that just didn't work out. Okay? Well, here is where the non-disjunction occurred on this one. But this one, we went through meiosis 1 and we divided normal. Right? We've got normal chromosomes there. And honestly, this left side is all normal. But now, during the second division, here's the second division, we had some non-disjunction, where they all went over to one cell and created trisomy and monosomy in that location. So it can happen in either meiosis 1 or meiosis 2. And as you can see in the zygote down here, that it's possible that even if there is non-disjunction, that it could create an individual that's normal, but it could also create extra or missing chromosomes. So females, we know, have 2 copies of the X chromosome and males have 1. So in female embryos, one X chromosome becomes inactivated. This is called bar body in each cell. That's just a little bit about how, so when you think of like an egg cell, it's going to have 2 X chromosomes. One's just going to be inactivated necessarily. And then you maybe heard that, before I move on to this side, like the male determines the sex of the essentially individual or fetus or whatever you want to call it, zygote. It's because all of the sperm are either X's or Y's, and all of the eggs are all X's. So if a sperm that's a Y meets an egg, it's going to be a male. If a sperm that's an X meets an egg, it's going to be a female. So that's why like the male determines the sex of the growing fetus. Okay, changes in chromosome number. So this is an example of Down syndrome. At the 21st location, you can see that the in that location there are 3 chromosomes. Remember, because it's a little off, because normally we have 23 pairs, but now we actually have an extra chromosome, and it can cause some deficiencies, but not necessarily inabilities in learning and things like that through Down syndrome. The other ones are called Turner syndrome, like we talked about. It's indicated by XO, and that O just means that there's not a second chromosome there. Somebody who has Turner syndrome would maybe be short in stature, just meaning kind of a shorter individual. They may have really broad shoulders, folds in the skin and neck, and also underdeveloped sex organs and breasts, which kind of makes sense if you think about it, because if you have one less sex chromosome, those are just going to underdevelop probably your sex organs. That makes sense to me, at least. The Klinefelter syndrome, this is when there's an extra X chromosome. So this is indicated by XXY. This is an underdeveloped sex organ and breast development. They may have large hands and long arms and legs if somebody has that type of syndrome. Here's a couple other situations, which would be a poly X female. An XXX tends to be tall and thin, but they do not usually have any mental disabilities. You may have XXXX, which may be a little bit more severe in that case. You have Jacob's syndrome, which is XYY. This is when that person may be really tall, may have persistent acne, may have some speech and reading problems if you have Jacob's syndrome, you as the hypothetical person. Changes in chromosome structure. Okay, so these next couple slides are going to tell us what happens when the chromosome itself has a change in structure that may cause some mutations or some genetic either differences. Okay, so the first one would be a deletion. This is known as like Williams syndrome. This is where there's a loss of a piece of chromosome, which makes sense. Like when you hit delete, when you're typing an essay and you're like delete, 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 that's essentially what's going on here. Okay, it's deleting a part of the chromosome. This would be, maybe then it would just lack traits or functions, or some things would just be a little bit off because your code would be a little bit off, right? So kind of when you have a glitch in your computer, right? There's probably a code that's off or a firewall that's not correct, all right? Translocation is kind of like the moving, like when you translocate for a job or an event. This is the movement of a chromosome segment from one to the other to another non-homologous chromosome. This is called the Alagille syndrome, which I'm not more familiar with that one, but essentially what I picture, and we have a picture in the next couple of slides here, of that movement of one part of the chromosome to the other. Just causes a little bit of kind of jumbling. Duplication is the presence of chromosome segment more than once. That makes sense when something is duplicated or made again. And then inversion, when something is inverted, like an inverted triangle. Inversion is where a segment of the chromosome is just inverted 180 degrees. So it just kind of spun around in a different way. So here's a couple of those pictures where we can have changes in chromosome structure. You have deletion. So on that far left picture, you can see that the little a was deleted, okay, which could cause some genetic disabilities or kind of things that are hard. Duplication. Now we have DEF. Oh, wait, we have DE, DEF. Okay, that's where you can see that duplication. Inversion. So you can see the D and the C over in that third picture, flip-flopped. And then translocation. The QR and the GH just kind of moved, right? The GH was on the left, the QR was on the right, and they just both flip-flopped. They translocated. Okay, so here's another showing how chromosome inversion happens, so here's that inverted segment, okay? When we go to cross over, remember crossing over was good, right? We wanted to cross over to have some genetic variation. But what happens here with chromosomal inversion is we're crossing over and then a mistake happens, okay? And this can happen at any time. Sometimes this happens and you don't even notice it. It's a certain type of mutation that didn't affect the function at all, okay? And if you want to take it to the next step, remember that table last week where we picked 3 of those letters and it would tell me what amino acid it was, like UGA was a stop codon. There are other stop codons that are very similar to UGA. So if I were to mess up and actually invert it and have a U instead of a G, it may not change the function. It may not change the amino acid. Sometimes it does, but not always. Okay. This is just showing the changes in the chromosome structure for deletion and translocation, just a little bit more in depth. Both of these figures can be found in your book on 1919, like the root beer, and 1920. All right, 54 slides later. Is there any questions about the material that we went over today? Okay, awesome. I think that we're kind of moving into bigger concepts, which is cool, right? We started really tiny. We started talking about atoms, and now we're talking about how chromosomes work and how like just switching a few things around can cause some different genetic diseases. It's also why genetics is such a really interesting topic, because it can be very applicable to real life. So that's all I have for you today. Enjoy your afternoon. This week we're supposed to have some good weather. Did you guys see that? It's supposed to be like 45 on Wednesday and Thursday, maybe even to the 50s this weekend, 57 next Monday. Get outside and enjoy it. Have a good afternoon. Get out inside and enjoy it, but don't forget about your assignment. Bye, thanks, you too.